In this screencast, we cover the hormones insulin and glucagon, and we will also discuss diabetes. Here are your learning objectives for the screencast. Describe how the pancreatic hormones maintain blood sugar homeostasis. Those pancreatic hormones are insulin and glucagon. And then lastly, contrast type 1 and type 2 diabetes. I thought we would start with a snapshot of the hormones that regulate blood glucose levels. The hormone that decreases or lowers blood glucose, what we would describe as being hypoglycemic, would be insulin, and it is secreted by the pancreas. Three other hormones are hyperglycemic, that is, they increase blood sugar levels. That would include the pancreatic hormone glucagon, and then cortisol, which of course is released by the adrenal cortex, which we discussed, and epinephrine, which, in addition to many other effects, also will enhance the levels of glucose in the blood. And epinephrine, of course, is secreted by the adrenal medulla. For this screencast, we're going to focus on the pancreatic hormones, insulin and glucagon. Both insulin and glucagon are secreted by the pancreas. The pancreas is a mixed organ. It contains both endocrine and exocrine glands. If you remember, exocrine glands release or secrete substances outside the body. Exocrine glands would include like the lacrimal glands that produce tears, salivary glands, as well as digestive system glands that secrete substances into the digestive tract. And the pancreas produces several secretions which it secretes into the small intestine. That would include bicarbonate to neutralize stomach acid, as well as a whole host of enzymes to digest various nutrients, which you'll learn about in A&P 2. In addition to exocrine glands, the pancreas also has endocrine glands, which secrete hormones into the bloodstream. The pancreas has two general types of endocrine cells. I assume they are named after the researcher who discovered them. They're referred to as aisles of Langerhans. You have the beta cells of the aisles of Langerhans, or simply referred to as beta cells, and they secrete insulin. And then you have the alpha cells, which produce the hormone glucagon. This image illustrates the stimulus and overall effect of insulin. So when blood sugar levels rise, beta cells in the pancreas detect that rise in blood sugar levels and secrete insulin. Insulin then targets the liver and causes the liver to take up glucose from the blood and synthesize glycogen. Remember that glycogen is the storage form of glucose in animals. Basically, it's animals' starch. Insulin also targets most other cells of the body and causes those cells to take up glucose as well. So as the liver takes up glucose and most other cells of the body take up glucose, blood glucose levels fall back toward the normal range. And as blood sugar levels fall back to the normal range, the stimulus that caused insulin to be secreted in the first place is reduced or completely removed. So classic negative feedback. Although we most closely associate insulin with blood glucose homeostasis, There are other stimuli for insulin secretion, and insulin also has other effects. Insulin can be released in response to elevated levels of amino acids in the blood, as well as fatty acids in the blood. And parasympathetic stimulation can also cause the beta cells to release glucose. Additional target cells for glucose would be adipose cells, where they promote the synthesis of triglycerides from fatty acids and glycerol, as well as skeletal muscle, where insulin promotes the synthesis of protein from amino acids and the synthesis of glycogen from glucose. I also want to add that there are some cells that do not require insulin in order to take up glucose or to use glucose for ATP synthesis. This will include the brain, the kidney, the liver, as well as red blood cells or erythrocytes. So not all tissues require insulin for absorption and utilization of glucose. So now let's look at how insulin promotes glucose uptake. So what you're looking at here in this image is this is the plasma membrane of a cell. This is the cytosol. So this is inside the cell, plasma membrane. This is the extracellular surface of that plasma membrane. So insulin comes from the blood, enters the extracellular fluid or interstitial fluid, and it binds to receptors because insulin is a polypeptide, so it cannot cross the plasma membrane. So it binds to insulin receptors on the outer surface of the plasma membrane, and it causes second messenger systems to promote the synthesis of a protein that is a glucose transporter. So that protein would be made by ribosomes, transported to the Golgi apparatus, placed in a vesicle, and then that vesicle would install those glucose transporters in the plasma membrane. And then glucose coming from the blood into the extracellular fluid can then be transported by facilitated diffusion from high glucose concentration to low glucose concentration. Glucose is charged. The only way it can enter the cell is through these glucose transporters or protein carriers for glucose. Insulin increases the number of these transporters in the plasma membrane, and that's how it stimulates the uptake of glucose from the extracellular fluid into the cell. Once in the cell, glucose can be used to make ATP and satisfy the metabolic needs of the cell. Up until now, whenever I shared a disease or abnormality that was due to hyposecretion or hypersecretion of a hormone, I told you you didn't have to understand the disease. I was only sharing details about the disease with you so you could better understand the function or effect of a hormone. For diabetes, I do want you to be able to describe both type 1 and type 2, and that's because diabetes is such a pervasive disease in our society. You likely know at least one person who has had or has diabetes. Diabetes mellitus is an abnormality associated with insulin. Diabetes mellitus literally means to run through sweet, and that's because someone with untreated diabetes tends to produce a lot of urine, and that urine tends to be sweet to the taste because there is glucose in that urine. Consequently, if they're producing a lot of urine, they will tend to be thirsty and drink a lot of water. So that water runs through them and comes out sweet, thus the name diabetes mellitus. And by the way, it ends in US. It is misspelled on your slides. In type 1 diabetes, there is typically destruction of the beta cells, or the beta cells have been damaged such that they do not make insulin, at least not normal amounts of insulin. So with type 1, you have hyposecretion of insulin. So in other words, the insulin is not there, or at least is not there at normal levels. So you don't get as many binding the receptors, and therefore you don't get the installation of the glucose transporters. Therefore, cells are unable to uptake glucose. Typically, this occurs in juveniles as a result of an autoimmune disease where, for whatever reason, the immune system decides to attack the beta cells of the pancreas. And this, in most cases, is genetic. So if someone in your family has type 1 diabetes, you are you're at high risk at type 1 diabetes. I do not mean to suggest that if your mom or dad has type 1 diabetes, that you're definitely going to get type 1 diabetes. That certainly is not the case, but the risk is higher. And typically this occurs because it's genetic, it occurs in juveniles. In type 2 diabetes, the beta cells are present and they produce insulin at normal levels, but the cells don't respond. So the insulin is there, it binds to the receptors, but you don't get your normal effect where you have installation of the glucose transporters. So what you typically find in people with type 2 diabetes, you find normal insulin levels, but also elevated glucose levels. So this is not an issue where you have insufficient insulin or hyposecretion. You have hypoactivity where the cells just don't respond normally to insulin binding the receptors. This historically has been referred to adult onset diabetes because you didn't tend to see this until someone was an adult. Some of the theories are that if you consume large amounts of sugary meals over time, the receptors become desensitized to the insulin. And so we typically didn't see that until adulthood. However, we're seeing type 2 diabetes at younger and younger ages, mainly as a result of childhood obesity. Obesity greatly increases your risk of type 2 diabetes. Now let's discuss the hormone that is antagonistic to insulin, and that is glucagon. The stimulus for the secretion of glucagon is a drop in blood sugar. If you're between meals, or if you haven't eaten for quite a while, there's little or no carbohydrate in your small intestine being absorbed. So blood glucose levels will tend to drop. That stimulates alpha cells of the pancreas to release glucagon, and glucagon is going to target the liver, causing the liver to break down glycogen. If you remember, that process is called glycogenolysis. So glycogen is broken down into glucose, and glucose is released into the blood. As glucose is released into the blood, it causes glucose levels to rise back to the normal levels. Now at the normal levels, the stimulus that caused glucagon to be secreted is reduced or removed, and so further glucagon secretion stops. Classic negative feedback. As was the case with insulin, there are additional stimuli for glucagon release and other effects of glucagon. High levels of amino acid in the blood, as well as sympathetic stimulation, increase the secretion of glucagon. The liver, in addition to breaking down glycogen to release glucose, also is stimulated to produce glucose from amino acids and fatty acids, as well as lactic acid, by the process known as gluconeogenesis, new glucose. And also, glucagon stimulates the transport of glucose out of liver cells. Glucagon targets adipocytes, that is fat cells, causing the breakdown of triglycerides for production of ATP, and also targets the intestines to enhance amino acid absorption. This illustration from your book does an excellent job of illustrating how glucagon and insulin regulate blood sugar levels. So let's say you are eating a jelly donut, or right after you're eating a jelly donut. Jelly donuts have a lot of carbohydrate in them. So once that jelly donut gets to your small intestine, glucose is going to be absorbed from your small intestine into the blood, and blood sugar levels are going to rise. That's going to cause the beta cells of the pancreas to release insulin. Insulin is going to stimulate the liver cells to take up glucose and store it as glycogen. That's going to serve to reduce blood sugar levels. Most other cells of the body are also going to take up glucose. As the liver and most other cells of the body take up glucose from the blood sugar levels are going to fall back to the normal levels. Now, let's say you are between meals, you haven't eaten for a while. Well, there's not going to be a lot of carbohydrate being absorbed from the small intestine into the blood. So blood glucose levels will start to fall. That'll be detected by alpha cells of the pancreas that will release glucagon. Glucagon will target the liver, causing it to break down the glycogen, glycogenolysis, breaking it down to glucose, which will then be released into the blood, and that will bring blood glucose levels back to normal. Remember that insulin is secreted when blood sugar levels rise, which is typically after you eat, and glucagon is released when blood sugar levels fall, which is usually between meals or when you're fasting. That is why insulin is often referred to as the hormone of feasting, and glucagon referred to as the hormone of fasting. To review, these are the learning objectives. Describe how the pancreatic hormones, insulin and glucagon, regulate blood homeostasis, and contrast type 1 and type 2 diabetes. I want to take this opportunity to remind you that for every hormone we have covered up to this point, you must know the following: the organ or tissue that releases the hormone, the location of that organ or tissue, the target organ or tissue of that hormone, what stimulates the release of that hormone, what its effects are, and its chemistry and mechanism of action. And then lastly, I want you to know if it's regulated by negative or positive feedback. And really, there's only one that we've covered that's regulated by positive feedback. All others are negative feedback. To help you organize this information in class, I provided you with these hormones and their properties tables. They include entry cells for all of the information you have to know for every hormone. Also, as I mentioned earlier, you can disregard progesterone, estrogen, and testosterone. You do not have to know the properties of the sex hormone.